and supported by the NOAA's Office of Ocean Exploration and Research, and the ship is operated by NOAA's Office of Marine Aviation Operations.
Okay.
This expedition is part of a 3-year campaign to address the Pacific Monument Science, Technology, and Ocean Needs called Capstone, that's its nickname, and it was designed to address fundamental needs of resource managers to better understand the deepest parts of the nation's marine monuments. So there are a lot of partners involved in this. NOAA's National Marine Fisheries, NOAA's Marine National Monuments Program, NOAA's Deep Sea Coral Research and Technology Program. Another important partnership for the Okeanos is the NOAA's National Center for Environmental Information. And aside from the exciting mission of exploration, the way Okeanos Explorer operates is unique in that all the operations are community-driven. That means the whole scientific community gets together and discusses what might be objectives for a particular series of dives. They are carried out on behalf of the entire science and management community, instead of being driven by individual principal investigators like Shirley Pomponi, our biology lead, or me, Patty Fryer, the geology lead. And through what you're seeing on screen, the telepresence that we're able to bring to you, you at home can see and hear right now this fantastic high-definition video. We're able to have a whole bunch of scientists on shore engaged in the expedition that we're mounting right now. More than we could ever fit on one normal research ship. So our scientists on shore are participating from exploration command centers. We call them ECCs, or, you know, just their office in their home institution. Also, important representatives from our academic, local, and state partners are involved in actually partaking in this expedition.
So for the ROV operations, we're partnering with engineers, videographers from the Global Foundation for Ocean Exploration. They operate and maintain these amazing vehicles, and they're bringing us the incredible imagery, video imagery that we're seeing here. And I would like them, since we just had a shift change, to please introduce themselves.
Okay, I'm Carl McCletchy. I'm the ROV pilot at the moment. On my left, we have
Sean Kennison, sitting navigator.
And on the right, Levi Unima as co-pilot.
Annie White as the videographer.
That's great.
Good. And anyone in the back row?
There's just Patty and me.
Okay.
Okay, thanks very much. Okay, so we have less than an hour to go for this dive. Go ahead, Patty. You want to go ahead?
Watch lead pilot. I wonder if, when you're ready, set up and stuff, if we could look at that light material exposed in the wall of the scarp. We're trying to determine whether this is reef material like what was seen on some of the earlier dives on the deep reefs.
Video, you can go in here on the light material.
Just trying to get a good look, folks. Seems a little better exposed here, so we are looking to see if we can see any shell fragments, bivalve shells, or anything that looks like ancient coral. Certainly the light color suggests this is not basalt or igneous rock, but rather limestone or coral reef deposits. The very white patch you see is the base of some stalked animal, but up the top of the screen, kind of at 11 o'clock, I'm wondering if that is also the base of something stalked. Kind of looks like it. Well, thank you, pilot. That was a good try. Didn't see what I was hoping to see, but
so nothing in there.
Nothing much. That's diagnostic anyway.
Okay, you can go wide video. So watch leads in general, are we just moving along this cliff, looking at the different corals and sponges?
We are. Chris Kelly, who proposed the dive, has suggested that we do try to document what is on this cliff, because the biology here is so numerous, rather than try to get to the ultimate final end point of the dive.
Okay. Well, we will
I think one thing we could do is we're kind of right in the corner of the wall right there, and it
Yeah,
might be sort of
what do you think
interesting
about,
to see if they can get into that curved
putting Sirius right
end
where
part
D2 is right now?
of
Just
the
kinda
wall
moving
right
down.
here, whether this community continues, whether it's orientation dependent, and that's why we're seeing so
See if
many
it just
corals
moves
here.
Sirius
I suspect we're going to see the corals right around that little turn in the wall
Got
coming
it.
up, but it just might be interesting to see it.
Yeah, 070.
Yep.
I think that was
Okay,
to get me
understood.
down off the cliff. But now that we're
Pilot, watch
in this
lead.
spot, I think it's
Stand by, guys. Go ahead, watch lead.
Okay, I'm sorry, Carl. What Chris would like us to be able to do is instead of heading upslope toward the end waypoint, try to follow contour around, as you can look at the quad screen, the curve in the contours on the slope to see if the biology persists or if there's something different about that curved area that affects the biology.
All right, we'll do that.
Thank you.
We'll get Sirius moving along the curve.
Yeah, so I was thinking about 2025.
But I'll just move Sirius directly over the wall, so we want to...
go a little to my starboard, so maybe like
I think
6-0.
Yeah, I think let's
6-0?
try a 6-0, yeah.
All right, just 20 meters.
Yeah, because we're fairly shallow, we can hop along. Yeah, if we had done 2-0,
Wow, there's a
then we would have to do
humongous
a
stalk
backup.
I
down
know,
there,
I
Shirley,
know,
on
I
the far
know.
right.
Saying that, I
That's
think
the
that's a
north.
Walteria, maybe a Walteria spot.
Splitting the
Oh,
difference
no,
is
it's
good.
gigantic.
Okay. I mean,
I mean, there is just so much. I mean, in terms of the abundance and diversity. But as you pointed out, no fishes.
Oh, I love this really delicate coral at the top.
There's some pretty large communities in here. Large.
Is that the same thing that Chris drew us
Yeah,
to
I think so.
previously?
I think so.
It's incredibly thin branches, tiny polyps.
video, would you zoom on this coral?
Sure thing.
We'll just have a minute while Sirius moves along.
That's partial.
You can go in full.
Okay.
There's a lot of it. That's good, thank you.
The pair of red dots you see are from
Okay,
our
over your clear
lasers,
video.
and they're 10 centimeters apart. Oh, that was a shrimp.
Can I get an establishing?
Oh, that shrimp seems to be interacting with the coral,
There you go.
Great,
I
thank you.
think. Playing with
Maybe.
the coral.
All right, I think we're good.
All right.
Well, Shirley,
have
the
a
thing
little
I
more
thought
time.
was a big branch. I don't think it is. I think it's
It's
a
like
crack.
a wet. Oh,
The sort
oh,
of
down at the,
gray
like
at the very
underneath.
bottom.
Oh, yeah.
I think it's a
Yeah,
cracked
could
surface.
be, could very well be.
Yeah, you
Yeah, because
see,
look
yeah.
at that crack is just coming right down to the right of that
Uh-huh,
big bush as well.
Uh-huh. Yep, there's a crack up in the top of the slope that penetrates the wall.
Okay, so geologically, what's happening here?
The cliff face is falling away.
Okay, that was
So-
a short response.
So the longer response is to the right, and because you can see the slope going down to the left. To the right, the upslope part
Is a good place to be.
is a better place to be. Yeah, the downslope part is sliding down toward the right, so you get cracks like this. But when we were driving along the edge of the wall, we saw cracks that were oriented parallel to the wall, meaning the
move along,
wall
Carl.
is falling off. There's a very steep slope beneath the ROV that is to the south east of our position right now. I'm not sure. Okay. Watch lead nav. Now I'm just wondering, are we waiting for the ship to move? Okay. We can,
we can move
yeah,
along
pilot,
this.
so can we move along to starboard a bit?
Yeah, we're waiting on the ship.
Okay. Okay. Couple minutes till we get more slack in the tether.
Yeah, I feel like I'm just starting to move.
Anything anybody wants to see in detail while we are here that we could zoom
There's a
on?
little nose over here.
Ooh, big block that has fallen off the cliff.
Well, we just got enough tether to see the next feature.
Whoa! Monsters. Coralline monsters of the deep.
I wonder if he could pan a little further to the right, just look along slope a little bit more.
Yep, we will be doing that, Chris. We're just getting a ship move in.
Yeah, pilots, the ship just stopped on that last move.
Excellent. Looks like Sirius will wind up in a good position.
Yeah.
I'll peek over the top of this and see what we have.
Come up just a little bit.
Yeah,
Oh my goodness. Look at this.
it's beautiful.
There are little corals all over the top of this and gigantic things hanging off the edge.
Very
Could
luscious.
see how thick that manganese crust is there, Patty, where that one part is completely exposed.
I do indeed. Yeah, this is outstanding, Chris. I'm beginning to wonder if the lumps that we see on the surface were not covering up, exposed boulders, but rather just were
You guys
parts
think
of
this is the
the
peak
thickness
curve?
of the crust itself and little areas where it seemed to grow
Now, do you want to
faster.
zoom my sonar out to 20 for a minute?
Yeah.
See what's
But
around us?
if this ledge that we're looking
sonor's
at
not seeing a
is
whole lot
all
right now.
manganese crust,
you could-
that has to be
keeps
a
going.
meter thick or more. Watch lead pilot.
Go ahead.
Wonder if we could have lasers on to try to get an idea how thick that upper layer is.
Lasers are on.
They are?
Yeah.
Whoa, where
the
are
water's,
they?
The water's just so clear that
Yeah,
you
can't
see
see the lasers.
what would you like them to be shown onto?
Well, I was just trying to get an estimate. You see that bright ledge from which that large branching coral is protruding. The material above it appears all to be just a thick layer of manganese, and I was wondering how thick it really is. It's the stuff that has the big white blotch on it in the upper right, that whole thickness.
Let's
If we
see.
could get a little closer so we get the lasers on it, where I can see them.
Yeah, there they are.
Here we go. Okay. So the little red dots in the center of the screen are 10 centimeters apart. So this is at least a meter thick right here. And that is truly amazing. It's much thinner up to the right. But it appears that there are portions of it where it's really variable in thickness. And what are these big corals that we're looking at, Shirley?
These look like these are the same. Let me go back. Ecnomyces, maybe?
Mm-hmm.
And then we can see that Iridogorgia, maybe.
The spiral.
Some other chrysogorgids. Don't know if that's a plexaurid down on the, like at about the 9 o'clock position. Whatever. This is a very, very diverse and abundant coral community for sure, and lots of sponges. Oh, here comes a shrimp. Tell you what, we're not seeing a lot in the water column, are we?
No, we're not. Occasional shrimp.
I mean, there's definitely got to be something in the water column for these corals to be surviving and growing as big as they are. So there are things that they're filtering out, absolutely, that they're suspension feeding on, for sure.
If you look out first to be.
Yeah, let me
copy.
Huge overhang here. Wow, look at this projecting out into space. Well, water in this case.
Looks to be what am I at?
Center just looks
6-0.
Nothing right there.
Yeah. Yeah, so we can move Sirius.
Yeah.
Yeah, just along the cliff would be a 6-0 again.
Copy. Let's do another 20 meters.
You guys agree?
Yeah. I agree.
Yeah, it looks great.
Okay, cool.
So I guess the question is, as we go around the edge of the curve of this huge scarp, whether currents change so as to affect the abundance of the animals here. Look at this block sticking out here. Isn't that amazing? I mean, you have to assume that that's the manganese crust.
Wonder what's underneath.
A lot of crinoids in
A lot of critters. Certainly crinoids hanging out
Love
down there.
looking underneath overhangs.
Oh, of course. But you don't want to put the ROV under it.
Video, you want to get a zoom under here?
Definitely.
Not really held by very much there. I imagine it's going to fall down fairly soon in the future, wouldn't you think?
Oh, I
Hopefully
would
not
think
real
so.
soon.
Yeah, yeah. Let it stay there for another hour or so.
Okay, so are all these small things, ribbons
Squat
on the
lobster.
rock, are they stoloniferous corals? I can't tell.
You mean the very tiny
Very
white
tiny white
projections?
things,
Let's go a
yeah.
little
Well, there
closer.
are some
Look at them
right
on
up
the
On the
corner.
surface,
Among-
yep, absolutely.
among
yep.
the
Right, so we can see that,
crinoids.
yep.
Yeah, it looks like.
Okay, that's good there.
That's just amazing.
That's not
Just
quite
amazing. They're just so coating
no,
the
no,
underside of this overhang here.
Yeah,
I
know,
agree.
I
Yeah,
know.
Scott, it's just phenomenal.
A couple different things that look like they're kind of on, well, they're not both on rhizomes, but it does look like there's stoloniferous octocorals.
Is that
and then
that little
something else,
carnivorous wormy thing?
The
upper right?
aplicophoran?
Yeah, aplicophoran.
I do not know. It's white, so I'm not sure. Different from the others that we've seen.
Okay. And underneath
Jeez.
there,
Then all that other little branching stuff,
Yeah.
I don't know if those are hydrozoans or bryozoans.
Yeah,
They have
that's
a little bit of a flattish appearance to me that suggests maybe bryozoans.
yeah, that's what I'm thinking too, Scott.
Can you go
Just a
little
feeling, you know, you
over
just sort of get the sense from seeing so
Yes,
much
see.
of this stuff, but don't know for certain.
Okay.
It's really nice to see these ribbons of octocoral polyps. So we've described these various corals that we're seeing as colonial, and the way that they're colonial is the original polyp that
Were they
forms out of the egg. There would be a larval stage, and the larva would turn into one of these polyps. They undergo asexual reproduction, basically a method of budding off a new individual next to them. And here you can see that they put out that little ribbon and remain connected to one another by the ribbon, and their digestive tracts would be connected through that ribbon. that's completely analogous to what's going on in the corals that have the large branching colonies, except that that tissue that you see here in the rock growing as a ribbon is completely surrounding the skeleton of these other ones.
Right.
Got it. Man, I am learning so much.
Les Watling said a decade or so ago, my friend Dave Malakoff wrote an article about Alvin and ROV diving, and he called it deep sea mountaineering, which is definitely what it is when you get on a wall like this.
Look at this projection.
Oh my gosh.
That thing is just going to flop right off. Oh, all that poor stuff underneath it.
I'll keep the ROV away for now.
That would be good.
Please do, Carl. Yes. Good idea. Carl said he's going to keep the ROV away from that.
We do have about 4.5
Wow.
tons to push down.
Oh, my, that's huge.
Is that how much D2 weighs? 4.5 tons?
Yes.
I don't think its payload is sufficient to bring that rock back.
No, we can only do about 40 pounds of rocks, 50 pounds of rocks.
Probably close to that today.
Yeah. Is there a big one in the port box?
There is. In the, no, I think just in the, we just have one.
Just
Starboard.
one rock.
Starboard, yeah.
But it is big.
It is big, yep. I'm just blown away at how big these corals are.
They have
oh
been
my
here
gosh.
a long time.
Long, long time.
Curious, though, as we go around this curve, we're at the northern apex. Well, I don't know if that's north. Is this north?
So it looks clear.
I don't know what projection we're looking at
Sidling
here.
along this way.
I don't think it's north. I
What's
think
our
it's
off-bottom time now?
Off bottom time is going to be,
northwest.
it's about, if we start to set up at 3:30, we'll be
Okay.
in good shape.
So we have a,
3:30 UTC.
so we have a little over 30
35
minutes.
minutes or so.
Yep, about 35 minutes.
We can definitely get around this curve. And there does
Wow,
seem to
look at
be
this.
less of the
There's
kind of large
corals.
things, or it's just less abundant here than it seemed to be a little bit further down.
So now we're seeing smaller things. There's some smaller,
Yeah.
smaller branching forms that we're more used to seeing. You've gotten spoiled by seeing all these gigantic
I have. I want them back.
fans. Well,
I
we're
like
rounding the
them.
corner. Looks like we might be seeing some more.
Okay.
Yeah, that just ends the last ship move pilots.
So the very fuzzy stuff that's
So I'm
on the
pretty
right-hand
square
side
here
at the
at
base
317.
bottom of the screen is the bryozoan.
Plus 90.
No,
Yeah,
the real small
Sorry to
ones
interrupt.
were.
Can we
Yeah,
get
go
a-
ahead. Yeah, go
Yeah.
ahead, Scott.
Yeah.
Can we get a quick zoom? It's on the lower left
Yeah.
as I'm seeing it. 10 seconds ago, I
Yeah.
don't know if it's out of frame, but it looks like it's almost looks like it was a whip, and then it had a spray of branches at the end of it.
Oh, so it'd be around
Yeah,
to...
Carl, can we look to the
Lyra,
left? Let's look over to the left. I think it's going
I4
off of the
whatever,
screen
I forgot
right
what
now.
plate it
Oh,
is.
oh, is it? Oh, okay. All right, then cancel that request. Thanks, Chris.
Oh, forget it.
Okay, never mind, pilot. Sorry,
Never mind?
never mind. Yeah, never mind.
Oh, I see what he meant.
Yeah, I would soon take a little more of a look down to the right along this wall, because I am really interested in seeing if the corals continue as the angle of the wall changes.
Sure.
Yep.
If you got a good look at it, that's fine. I didn't think it was the lyrate form, so that's fine. Thanks.
Like looking
Seems
down
to
on this,
be
it reminds me
a-
of like fireworks.
Seems to be a real difference in the rocks to the left and
Yeah,
the right.
looks-
There's
yeah.
a
Kind of more
demarcation
angular.
between what may be a fault and then all of this rubble here, much rougher seafloor, and again you see more animals on it. So that block may have broken off more recently than the material right here. But it's hard to tell.
So we're looking down on different chrysogorgids. Is that what we're seeing? Some metallogorgia, Boy, maybe in a Riddick Gorge. I don't know.
Oh, one. Go ahead.
No, you go ahead.
I was going to say the one thing that's very striking to me is how cleanly cut this manganese crust is. These faces are
Fish.
so...
Oh, pilot, we need to look at the fish.
Yeah, I see the fish. Let's go for the fish.
And it looks like it might be holding still.
Yeah, for now.
For now, yes.
You know what happens when the
Creep
giant predator
on it.
comes up to a
Creep on
fish.
it. Creep on it very,
video.
very slowly.
Zoom in.
And quietly. Do not
Very,
use any
very
noise.
slowly.
Oh,
We want to
pretty
see
loud.
all of its morphology.
Okay, so
Beautiful
beautiful.
scales.
This is a different fish.
Yeah.
Can go in a little tighter video.
And we're looking at the top of it. It's lying on its side,
It's asleep, I
sleeping, has big eyes.
I have a little bit.
So I think this is what's called a slickhead and a lipocephalid, but I'll double check right now. We just don't come across them that often.
Okay, you want to come in a little tighter?
It has something
Right,
floating
pulsing.
near it.
Okay.
It does have something on its fin.
Yeah, it looks like it has parasites on it.
Yep, its fin has a white blob on it. The parasites are diagnostic, so I understand,
Kind of
for
drift
some
down into
species.
its tail.
And maybe we'll
Okay,
get to
can
see
come
them. Yes, it
outside.
does have parasites on its fins.
That's good.
Looks like
Oh,
barnacles.
can we zoom on those parasites?
Yes.
Ooh,
Zoom
yes.
full center.
As we were told, these can be diagnostic of the species sometimes. This looks like amphipods. And the gill structures
Okay,
are
come out of
critically
that. It's
important,
starting to get agitated here.
right?
That's correct.
Hmm.
Looking at my little notes here.
Well, we-
Detail of the head, gill cover,
Try to
eye,
get-
mouth region, an area of the body immediately behind the head, including the pectoral fin.
Well, this one is
So these
cooperating.
are kind of interesting because they have
Perfect.
somewhat close to the body form of many shallow water
Pilot, can
fishes,
we-
unlike a lot of the stuff we see down here, which is more eel-like in appearance. So
That's cool.
and I don't really know enough about these things to really understand where they are in the teleos tree, but it's a pretty interesting fish.
Oh, that's really good.
Okay, you can come out of touch.
Well,
Zooming in
Ken
there.
Sulak, if you're watching or listening, I hope you're liking what you're seeing here.
There's
Comment
the
on the head
mouth. Mouth is important. And the gills. And the area immediately behind the gill. I don't know how anyone could ask for anything more. This is great.
It is great. Carl, once again, you're amazing us. Oh, lateral
Well,
line,
he is
yeah.
kind of sleeping, so.
This little fish
Oh, look
is
at all that.
not feeling energetic.
Okay, let's try
Well,
to get
hopefully he just
parasite
ate
here.
something.
Yep.
You go in full on that.
And not that he's feeling the ill effects of carrying amphipods about with him.
Is that what they are?
I think so.
I don't know.
Okay, let's
I don't
try to
know.
get
Are
a
those
whole body with
amphipods,
lasers,
people?
and then-
Asaka Matsumoto says the growth line and the scales are visible. Excellent close-up view. Scott France thinks these are isopod parasites.
Isopods, not
Yeah, I'm pretty
amphipods.
certain those are isopods. And if they are the ones that belonging to the family that I think, they're not permanent parasites, so they can feed on this fish for a while, then swim off and find another one.
There we go.
Nice job.
Thanks,
I
Scott.
turned to keep you in view a little bit.
Okay.
And I have a nice sonar return of another wall. All right, as a like maybe the end of the.
Yeah.
End of
Wow.
the canyon.
Yeah.
Yeah, it does rise over there.
So we can go back
Good
down.
video of this fish.
Yep,
Okay.
and shipped its up a couple minutes ago too. So
Okay.
probably just a little more swing of serious. That should be good.
Start coming back down. Reacquire the edge there.
Should I come back out?
Yes, please. Thank you.
Full wide.
So we're about
That was great. Thank you.
22.5
Thanks very much, pilot and video.
meters above the seafloor. This gives you a sense for, what it's like in terms of scale of the features that we're looking at. If you look at the Sirius view as we settle back down, you'll be able to see how incredibly impressive the scarp is that we're approaching. Just measuring from the bathymetry, we saw higher than 80-degree slopes on parts of the scarp, almost completely
25.
vertical. And what we have seen during the dive is
Yeah, there's no
overhangs.
danger of hitting that. We didn't move the ship far enough.
Yeah, I was just wondering if it's what I see in the upper right of my screen. Upper right of your screen.
Looking down at 65. See the
Okay.
see where the tether's at?
Yeah.
Okay.
Okay, I'll keep sidling to the right then. Until you're back to, you're at about 30 degrees, right, co-pilot?
Yep.
Oh, wow. Big sponges, Shirley. Are these the encrusting kind? Or not
No,
encrusting,
no, you
but
know,
they
you can.
have the
No, they're
little
fan, and you see the.
spicules?
No, look at the crinoids on the underside of it again. These are like the ones we saw
Oh,
earlier.
right. Oh, right.
I'm back at 30.
Oh, there are crinoids all over all of these. But only on one side. How come?
Taking advantage of the current.
Well, the upper one, they're on the underside. The lower one, they're on the upper side. So, how come?
No say.
Maybe they are both on the rougher side of the sponge.
Maybe.
Oh, thank you. That makes sense.
Maybe.
That's easier to grasp with their Siri.
Come down a little bit.
Okay.
Not too much. Trying to keep slack out of the tether.
He is gonna gain down.
So the next wall starts about
Wow, a lot of those sponges
15
here.
meters
This
to
is
my right.
different. Different collection of organisms here. Many more sponges.
Big white pad over there.
Yeah, it's big.
Yeah, so far we're getting more of these big sponges and fewer of the
Of the larger.
larger
Yeah.
corals.
Yeah. What's that?
What's
The distance.
that? Is a huge
Oh my God! What is that? What
It's
in
a
the heck is that?
gigantic.
No, wait. Oh my God! What is that?
A
Well, we'll
sponge.
go there. Yeah, it's definitely a sponge.
All right, I have to like turn off the mic.
Yeah,
Seem
she's
much
going to
bigger.
screech, folks.
Oh
She's like,
my
wow.
God! What is that?
It's a holy sponge.
Oh my God! All right,
Oh,
I'm standing
yeah.
up now. What the heck?
So this is the other cliff, basically.
Yeah.
Uh-huh.
So it goes like that.
Yep.
So now if you want to drop a target, I don't know, new wall or north-south wall or something. It's much taller too.
You know what?
Chris and I are being silent.
Okay, while you're being silent, notice, folks, that there is a huge
wall.
wall,
Oh
and
my
there's
God.
a giant fissure between the wall that the sponge is on and the rocks just
Holy
downslope
smokes,
to the
what
left.
the heck is
There's
that?
a big break here. Maybe there's, more concentration of current through this area. But this is a big sponge.
Okay, how big do you think that is?
Sponge, believe it or not, is in the genus Poliopogon again, and I just can't remember what the letter designation is. It's characterized by these really huge holes that run through it that's unlike any of the other Poliopogon or Poliopogon.
So it's a glass sponge.
Family Pheronematidae.
Jeez
And we
Louise.
used to call this thing sponge holy when we first saw it because we had no clue either what it was. And then it was eventually sampled and examined microscopically, and we found that it's in that same genus, but quite different.
All right, so I'm trying to estimate how big it is.
Oh, do we have the lasers
20.
on?
Yes.
We do.
Yeah.
Whoa, where are they?
I'm thinking
Big.
I'm thinking it's got to be at least
Can do
a
little zoom on
meter,
Sirius there.
Yeah.
at least a meter across. Oh my God. Okay, Scott, I'm going to have to say it. It is a jump squiffling sponge,
There are the lasers.
which means very large.
jump spiffling.
And it's also a wopsy whiffling, jump squiffling sponge. Super awesome, very large sponge.
Super awesome, very big, yeah.
Oh my gosh.
The red
Do you wanna
dots
go
are
in
10
tight?
centimeters apart.
Right.
It's about 4 inches, so now you can get an idea how big
Very
this
close up with
is.
the structure
Oh
here.
my gosh.
Wow. I'm surprised there aren't more shrimp, squat lobsters running around inside
Right,
of it.
exactly. Now,
Why
how
aren't
old
there?
might something like this be?
Pretty old, kiddo.
How old?
To get that big.
Oh my gosh. Of course, the
Well, look at
question
this.
is, how
Look,
fast
look,
do they
look,
grow?
look.
Yeah.
Look at- oh,
look
wow.
at right. Yeah, there you go.
Serious
Look at the
view.
serious view.
Is phenomenal. Okay,
So-
the ROV, I'm told, is about 9 feet tall.
How wide?
How wide is the ROV, guys? Pilots?
6 feet?
Yeah, probably about
About
6
6
feet?
feet. So,
Whoa, so this is like from the top right to the lower left, it looks like it could be 2 meters long.
Okay, whenever you're
2
happy
meters
with
wide.
that video, you can come
We can
back
tilt
wide.
the ROV so we can measure it.
don't get too close.
I know.
Oh my gosh.
That is huge.
Is that full wide?
Chris, go
Sorry,
ahead.
my
Can you go ahead and
microphone.
type in the name for that, please? You're saying it's a pheronematidae?
Yeah, I went and punched it in. It's Poliopogon species F.
Okay, I
is
see
what
it.
it's known
Okay, I
as.
got it.
That'll give you
There
an
we
idea
go.
of how many unknown species of Poliopogon there are.
I'll tell you what, we're going to have a hard time today picking out the
Yeah.
2 images for our dive summary that
You
have
bet.
to be the highlights. Oh my gosh.
Okay, you can go wide on Sirius now.
All right. This is amazing. And look at all the crinoids on the inside.
That looks like the wall's 25 meters from me
Okay.
Jeez.
at its closest point.
Alrighty.
So can we look a little more along the wall, or
We will,
are we
yes.
too close to the end of the dive?
No, we're good. We have about
Yes.
20 minutes left, Chris.
Yep.
Okay, well, let's go ahead and pilot and continue on the wall, please. Thank you very much.
Say again, video.
She good?
All right, guys, we have a new direction now. Straight up the wall.
Up the wall or along the wall?
Well, we need to find the top.
Ah, the
Let's
fuzzy
go along.
things are these, the bryozoa?
'Cause there's overhangs.
Yeah. Yeah, I cannot see over it.
Maybe.
Yeah. But we can.
Ooh,
I don't
maybe
think
do
I don't
10
think these are the
meters
ones though
closer, or-
that Scott was
No,
talking about.
I
The
don't think so.
bryozoa.
These are-
No,
these
these
are
are not
much
the ones.
bigger.
10 meters closer.
Oh,
Yeah.
yeah. No, the ones that we were talking about were kind of more of a brownish color.
No,
I
keep
meant the
cycling
things that
over
were
to some
under the
different
overhang.
direction.
Ooh,
50,
there's
10
a
meters.
stalked crinoid, the red thing?
I'll put it up.
Is that a red?
Is that too close?
He looks.
What are you,
Yeah,
20
it might
away?
be. Yeah,
25.
might be.
I have enough now. It's,
We can just come up for a bit.
yeah, I can come up for a bit. Might get uncomfortably close if we do 10.
Yeah.
So all of those sponges that we saw just recently were closer to places where the rock wall has vertical fissures running through it. And I'm just wondering if that does tend to concentrate the current.
I think I'm starting to see over it.
This has certainly been another interesting dive.
It's so changeable.
I don't know, Chris, did you have any idea that there would be this much here? I mean, you know, you had thought that there might be sponges and corals. Did you have any idea it was going to be this much, this abundant?
I stopped the winch.
Well, we had some sites on ridges last year that were really high-density sites as well, so I'm ever hopeful. But, yeah, I mean, you never know until you get down.
Right.
And I think Derek's multi-beam last night really helped out because we didn't realize how steep this wall was until we saw it this morning. And it made perfect sense to kind of go over to the edge of that wall and take a look because Scott has perhaps more experience than I do on these really steep walls like that. He's found a lot of corals in this type of terrain.
Yeah, we're really fortunate because we have an excellent mapping team, expertise, capabilities to be able to, in some cases, get multi-beam imagery that we've, you know, of areas that have never been mapped before, and some we just have not very fine scale. And so what our mapping team's been able to do the night before a dive is to go in and get some more detailed mapping, and it's helped us to refine our dive tracks on many days.
Well, you know, it's not just that they collect the data, but they process
Oh,
the
yeah,
data.
they process it,
They
yeah.
produce the map. They interpret the map in terms of slope and everything else that we have been taking advantage of here in the minutes before the pre-dive call. It's just amazing how much effort that they put in. And it's greatly appreciated, guys.
Thank you, pilot. As soon as video has enough beauty shot on this, because this is awfully pretty, we're good.
All right, I think video's good.
Okay.
And we're full wide.
We are at 1,720 meters.
10-meter move now.
All right, 10 meters at 50. So fine.
I'm good with that, yeah. I think I see something large and white.
Okay, I see that.
Okay. Yeah,
Many
sounds good
fewer
now.
critters on this wall.
Yeah, putting it in.
This wall is also a lot rougher and appears to have a thinner manganese coating. It looks like a wall of little fern.
It does.
Yeah, angel hair fern. Here's a sponge.
To your port side too.
One of these
beautiful.
vase-type sponges.
Probably the one that has that kind of a euplectellid type mesh on the top. All of these beautiful fans sticking out. We are coming up this wall.
Holy
Wow.
smoke.
Big.
Oh, monster sponge.
That I believe that's a paddle sponge loaded with crinoids.
Yep. This is a crinoid condo. Wow.
See, now when you look at the stalk of that paddle sponge, that also reminds us, if it weren't so curved, or it does remind us also some of these
And pilots and Washington, we have 15
stalks
minutes left in the dive.
of the dead sponges. Roger that, thank you. Roger that, thank you. Okay, 15 minutes left in our dive. And for our shore-based scientists, we will have our dive planning meeting for tomorrow's dive within about 5 minutes after we come off of the bottom. So we have about 15 minutes left in this dive. So in about 20 minutes, we will have our planning call for tomorrow's dive, which I think is on Subducting Geo 2.
Right.
Deep dive, but an 8-hour dive.
Those are forests, and those are little trees.
And we hope we'll be looking at the interior of one of these geos, possibly with less covering of manganese, so we would be able to see reef deposits if that's what's exposed there.
Big sponge down here. With a nice little
Ooh,
overhang
what is
line
this
there.
sponge, Shirley?
Yep.
It looks like a lily, the surface of it, top of it.
It does, doesn't it? It's a hexactinellid sponge. Not sure which one. I'm sure Chris knows.
There's another giant.
One. Yeah, another.
Oh, look at the image in the
Yeah,
Sirius
yeah,
view. This
yeah,
is a big
yeah,
one too.
this
Not
is
as
really-
big, but
No,
wow.
but pretty big.
So Shirley, you can see for this view that it's got the fuzzy, hairy.
Yeah.
That's going to put it in aromatid.
Right. Oh boy. So pilot watch lead.
Go ahead.
I wonder if we can zoom in on the base of this sponge to see those hairy attachment features.
Oh, well, it's.
No, we don't need to. We can see it from here.
I can see it from here, so that's really no need. But I was just pointing
Relay
it out
that.
for Shirley so she could
Right.
see the similarity and why it's in that family.
Okay.
So I have a feeling that the family is going to be revised even after these Poliopogon get described, because I suspect that they're going to wind up getting ultimately split into two different genera. They just have a lot of different morphologies. And this is a perfect example of one that just doesn't look like any of the others. So right now it's being tentatively put in that genus, but I'll bet it doesn't have, I'll bet it's not going to be there for too long.
And you'll pardon me for changing the conversation from sponge, but I just have to remark on this wall again, the density of these Chrysogorgiidae colonies, which may be Pleurogorgia, are so reminiscent of the wall of Karen Ridge that we climbed up last year. And on that wall, if I recall, we went about 100 meters in vertical elevation and just continued to see hundreds and hundreds of these colonies. And so I wonder if we were to do the same thing going down this wall, if we would continue to see these colonies all the way down there. Just incredible densities, which speaks to how much food must be passing along this wall.
Yeah.
Yeah, I don't see anything on sonar that should bump us. Do you just want to do 20 meters at,
For those of you joining us from
you said
home,
135?
tomorrow's dive is going to be a little bit deeper. We are going to be starting at about almost 4,400 meters. So it's going to take us about 2 hours and 45 minutes to get down to the bottom. So if you want to join us on tomorrow's dive, we will be on the bottom at around 11:15 a.m. ship time. In Guam, that'll be 9:15 a.m. Hawaii, 1:15 p.m.
If it's a short move,
On the west coast of the U.S., that'll be 4:15 p.m. And Scott, where
so 20
you
meters
are,
at
it'll be around 6:15 p.m. And Deb, where you and Nolan are, and others joining us from the East Coast, Tara, it'll be 7:15 p.m. And for all of you viewers from home, we hope you will join us tomorrow as well. We still have about 10 more minutes left on this dive. Still seeing incredible abundance and diversity of organisms along this manganese encrusted
slope of a geo.
We're looking at a scarp.
We are looking at a scarp right now, but the whole thing
I know,
is
it's
on
very
the
cool.
slope of a geo.
I know, very cool. Very, very cool.
Oh wow, look at the surface of this. Oh, just rolling hills of manganese oxide crust with tiny little
Come around a
corals
little to
resting on their tops.
split the difference a little.
Copy.
Don't you think before we leave, we ought to zoom in on that and just get a nice view
Of the fans?
the fuzzy wall?
Sure, that would be great. We'd love to do that. Pilot, we would like to zoom in on all of those beautiful fans that are
projecting from the wall?
Right, all over the wall.
All right. I think I can find some.
Yeah, pick one, anyone.
Okay,
Get them
video.
all.
We'll just,
Get them all.
get
Kind
a
of
look at all
zoom
these
and
fans.
sweep.
Maybe go in just till you can't see the
Oh,
lasers.
pretty,
They're
pretty.
so delicate.
So this is what we have been seeing all along this wall.
I'll try to go
Oh
slowly
my
along.
gosh.
Little starbursts on arms. Just hundreds of these.
Thousands. Millions.
Shirley is channeling
So I
Carl
guess
Sagan.
one of the values of this video is hopefully it'll bring to a lot of people's attention that there are some pretty amazing communities down here that are living on these crusts. It's not a barren area where you could just go do whatever you want and not jeopardize resources like this. And so I'm absolutely delighted about what we found here today and some of the imagery, because I sure would like to make sure that that message gets out, that as deep sea mining for crusts becomes closer and closer to reality, the process needs to happen very carefully, and site selection needs to happen very, very carefully as well, because you can see on this dive what's potentially at risk.
I know people tend to think that there's not much life in the deep sea, and certainly on the dives that we've made during this leg of our expedition and the first leg as well, we've been able to show that, in fact, in many, many of these areas, which is just a very small fraction of the total bottom in this whole National Monument area, we are, in fact, seeing an abundance and diversity of life. Invertebrates, fish, just really, really incredible. And as Chris mentioned, some with commercial importance. Precious corals, the fisheries, the non-living resources, the minerals, and so on. So really important to be able to document these communities so that the monuments resource managers and the relevant fisheries management councils are able to wisely manage these resources. And here we are at the top now of this ridge, getting close to the top. We are at, where are we? I don't see it.
Depth.
No, I know we
Depth is
got
not displayed at the moment.
I'm saying depth of Sirius. 1708. Thank you, Carl. So, Scott, the fans that we saw that were in such high density, they were the Pleurogorgia. Is that what we were looking at?
That's correct, Shirley. Pleurogorgia is our best guess at the genus, and it's probably correct.
Another big
Thank
sponge.
you. Yeah, another big sponge. This one looks different.
It is quite different, yes.
We're just passing on the left on the upper surface a Metallogorgia juvenile, and I think that's the first Metallogorgia we've seen today. Oh, no.
No, we saw, we saw one earlier.
But it was mature.
Yeah.
Want to do a half zoom on Sirius? It's a nice image there.
Nice.
Okay, beautiful view of Sirius, from Sirius of D2. So you can see the Deep Discoverer from the Sirius vehicle. You can see that fuzziness on the wall. Those are all of those fans of the Chrysogorgia, the Pleurogorgia.
And just below the ROV, you can see the flat surface of a block of material that has slid down this wall, is resting slightly below the depth of the upper surface. So this is how these walls form. one block at a time, just breaking free and sliding down.
Yes, video, can you zoom on the sponge?
Of course. That's a partial.
Yep. Very nice. And again, Chris, you can see those spicule brushes that are attaching the sponge to the surface of this rock. They've got to have strong little grippers on the the bases of those spicule fibers.
Super Velcro.
Super Velcro indeed.
Okay, and you can go in tight if you'd like.
Oh,
video.
wow. Spicules all over the surface of
Structure
that
looks
thing.
pretty nice.
Ouch.
That's full wide or full
Ah.
zoom.
Nice spicule fringe. Beautiful. Very nice mesh on the surface. Great. Thank you, Pilot. Whenever video is finished getting beautiful imagery, you can go wide.
Oop, jelly just went past Sirius.
All right, I think I'm good whenever.
Okay. Go what?
Oh, and there was a
Bubble
corrallid. Yeah, corrallid just to the left, and there's one. I'm not sure if that's one just to the right.
looks similar.
Okay, can we go wide on Sirius
Chrysogorgid.
also?
Yep.
Oh, and then looking up on the surface there, we can see in the distance some Walterias, some more Chrysogorgids.
An incredible abundance.
Oh my gosh, yeah.
Many more than
All the
I
octocorals.
had expected
Yeah,
to see.
I have to say, me too. This was really surprising. And I'm looking in the distance there. Maybe those are two Metallogorgias all the way in the distance as well.
Oh, look at these. Yes, those do look like Metallogorgia. And look at all the stalks right below this little ledge,
Yep.
indicating that this block has just dropped
It's like
down
a little
in
dump
front of
versus
us.
forest here.
Yeah, I was
That's
just thinking
only a
about the
meter
truffula
high
trees.
or so, but it seems that's enough to disturb the animals
There's a couple
sufficiently
in
that
that look like truffula trees.
they have a problem. And a lot of stumps right on the top of this ledge.
All right, Pilots, in 1 minute, and then you guys need to start to set up to get off bottom.
Okay, we have a
Sigh.
minute to go.
I
This
don't
is a
wanna go.
beautiful vista that we're
let's
looking at
stay
to
here.
just remind us of this this dive and how much we've seen in terms of biological diversity on this dive.
Shirley,
Yeah, go ahead, Scott.
Shirley, I was just going to say we're looking on a side view of, in the center, two Metallogorgia colonies. The long stalk with it looks like an umbrella at the very top. And one thing I find interesting is neither of them are tipped over,
this
which
is video.
suggests that the current up there
Looks like
is actually
there's a
quite
jelly or something in the
low
water
because
column off
usually
the
they bend with the
right
current. And then the one on the left looks like it still has a couple of branchlets underneath the main top parasol, which
What's
is
going
what
on
they
this
have
coral
when they're in their,
that's
quotes, teenage years as they're growing up.
You going on this coral?
And I think I see a swimming isopod kind of swim walking through the water column to the right of the Metallogorgia.
Thanks, Scott. As always, we appreciate your eagle eye.
Okay, you can go
All right,
ahead.
Pilot, take your last
the
zooms.
Fascinating
breadth
commentary.
breadth and depth of knowledge about all things invertebrate and
Coming up for you.
wow. Wow. Look at that.
And that's not quite full zoom.
That's okay. I'm bouncing a little out here.
Yeah.
Great. Thank you, Pilot.
Thank you. Unfortunately, I think it's time to leave the bottom.
Time to leave.
Yeah. Okay, whenever you're done, video.
I'm not sure I
Yeah,
saw
5 more
the
minutes, Mom.
isopod.
Oh,
I'm
this
not
one.
sure.
Can we stay 5 more
I'm not
minutes?
sure I saw the isopod, but yeah. Oh, is that what all those things are swimming?
Let's check it out. Okay, I want to zoom on the water column here.
Yeah, that'd be great.
There's so
Go
many
up a little
corals in
bit,
the
yeah.
background.
Oh gosh.
That one doesn't look like
No.
anything.
No, it doesn't.
There was one that was moving.
Yeah, it looked
Should
like
I come back out?
Yeah, come back out partial.
Okay, well,
I don't
that's
see anything. Okay.
that's good. Thank you. Thanks
All
very
right,
much.
Come wide video.
Okay.
Okay.
Thank you.
So it seems to me that there's a lot more animals on
All right,
the
so I
top of the
starboard
ledge
a
at
little
this
bit.
particular location, right in the crook
Looks like if you turn
of
30
this
degrees to starboard,
area
you'll
here.
turn
So
this
I
way.
wonder if there's some general current
Has the,
acceleration up through
pool
this little
time
canyon
put in?
head feature
Uh,
that's supporting a larger
Dan still wanna do that?
population on top here.
Yeah, and we're seeing a lot of things in the water column too. Yeah,
Dense up here.
for sure. Oh yeah, now we're seeing
We
some
talked
things.
about
Great.
it, I think, today.
All right, folks, thank you very much. We are at the end of our dive on
There's something
Vote
right in the middle of the
Guyot
water column.
and
Guyot.
Guyot,
Look
Vote
at the
Guyot,
shrimp there.
silent G, silent T.
Yeah, we gotta go.
Yeah.
And we hope that you will join us tomorrow as we make a dive on the
right.
subducting
I'll, slide over.
Guyot 2. And that'll be a little bit
Starboard,
later because it'll take us about 2 hours
See
and
if we get your
45
turn
minutes
out.
to get down
Okay.
to the bottom. So
Without doing any math.
we'll be on the bottom at about say 39, 13, 11:15 ship time. So we're UTC plus 12. So calculate that out for your time
watch
zone. And we really
Yeah,
appreciate you
watch out.
joining us.
Don't forget to turn yourself.
My name is Shirley Pomponi, and
You
from
put in auto depth.
Harbor Branch Oceanographic Institute at Florida Atlantic University, biology science lead. Wait.
Lead is Geology Science
Not
Lead
getting
is
anyone at
Patty
workshop.
Fryer from University of
It's okay, I
Hawaii,
think
Hawaii
last I talked to
Institute
him, he
of
wanted to do it.
Geophysics and
He figured out which
Planetology,
way
and we
Jack has to
will
unwrap the
see
tether
you
and all.
tomorrow.
So on behalf of the two of
Alright.
us and the many
So,
scientists
I want to
shore-based
have
who
0
have
wraps,
participated in this
but
dive,
then-
we'll see you
So that
tomorrow.
would be a turn
And
to-
shore-based scientists, stick around. We'll be with you in just a couple of minutes for
starboard.
our
Okay.
dive planning
0
call for
wraps,
tomorrow.
correct?
Bye-bye,
Yes.
everybody.
But